All right. So as usual, if you have if there will be any problem along the way that anything is accidentally cut out, you can come back using the same link. Okay. So what I plan to do today is just give you a lecture like throughout the 1.5 hour, and then maybe we finish before that, and if we have time at the end, maybe you can ask me question. I don't limit to any question as long as it related to deciding of your research. Okay, so just let's start. Hold on. Okay, just okay. I hope everyone see this all right. Right. Yesterday we finished off with this slide about the overall picture of the research design. Okay, and I always have problem with the first slide. Okay. Okay. It's actually this slide that we finishing off. So today, what I'm going to talk about is all the aspect of the technical design. Okay, three of them in technical design, like I told you earlier, that it involves research strategies, research materials, and research planning. Okay, the first element of research design is research strategies. So the research strategy is basically the way we plan to achieve our research objective. Like I told you earlier, that when you designing your research, you start off with your conceptual design and you should come up with the objective of your research. So research strategies is various way that you do to achieve that objective. So you have to bear that in mind. Okay, what objective you have, and what experimental plan you are going to do to achieve that objective. Talking about research strategies, there are so many ways that you can achieve your experiment or your research results. Okay, so for each of the experiment, you might have different strategies. I mean in your research. Okay, in your research, you could have different strategies that involve. Is may not be only one type of strategies. Okay, so when we are talking about strategies, approaching of each strategies can be seen in two ways. So key decision in selecting the strategies that you want would depend on whether you want the results that you get to be like depth or breadth. Depth means you go deep, understand maybe only one topic, but deeply. Okay. Or breadth means if you are interested in so many points, so many topics, and each of them might not be so deep. So that's depth versus breadth. So knowing what you want would knowing what you want is really helps in selecting the strategies. Another thing that you have to think about when selecting strategies is that whether you want your work to be qualitative or quantitative, right? You want the exact number. If you want exact number, it has to be quantitative. But if you just want like if you just want like whether just to tell the level plus or minus or not exactly having numbers with it, it could be qualitative. Another thing that you have to think about is whether you want your experiment, okay, to be empirical or it's a desk research. Okay, so empirical means that you are doing experiment basically, and it is sometimes referred to as a primary research. That means that you hands-on the experiment, you get the fact, you get the results from that, and that is where you get your data from. Another, as opposed to empirical work, is the desk research, which is known also as a secondary research. So you search for your data from whatever has already been published. Okay. So that is the decision that you have to make when you are selecting strategies, whether you want a deep or wide aspect. You want it qualitative or quantitative, or whether this work that you do have to do the experiment, hands-on work, or it can be done on this research. Okay. And I list out, okay, the strategies that normally used in research project. Okay, so we might be familiar only with experiment, okay, because for us, when we are doing research, we are talking about, okay, we have to do experiment, but it's not always that way. Okay, I want you to be aware of this sort of stuff as well, because there are so many method or many strategies that you can use to do your research. Okay, the first strategies that can be used in research is survey. Okay. Survey, maybe you have been subject of surveying, like if you walk somewhere and then people approach you and then ask you to do the questionnaire, that is a part of survey research. Or for some field, they probably do like field survey. So like geology and those type of field, they might go somewhere and then do the survey and do something about their research using the data that they obtain from surveying. Okay. Second strategy is experiment. So this is very common to us. Everybody knows what experiment is. You plan experiment, you hands-on doing experiment, taking samples, analyzing the samples, and then getting the data from whatever you do. Okay. Another type of strategies is the case studies. Okay, the case study is really popular right now because if you think of it as like during this COVID period, okay, so people start getting vaccination, people they having a lot of data available, and there's a lot of case available. So the case studies, what I can think of as a most common field that use case study is probably medicine. I mean, if you involve the patients, if you involve this type of thing, and you're collecting data from them, so that is the case studies. That is one strategy of doing research as well. Okay, so they don't have to actually do the bench work experiment. They can do something like that. And I can tell you, like for the case studies or those type of things, they have to involve a lot of good statistic work. Okay, because when it's a case study, you have so many variables, you have to choose a correct statistic, and you have to interpret the data statistically and correctly as well. Okay. Another strategy in research project, okay, is the ground theory approach. Okay, the ground theory approach. So so with this type of research is sort of you have to, if you are interested in something and if you can't really hands-on the experiment, okay, you could think about the theory that possible involved and try to obtain something from the ground theory to prove the thing that you are interested in. Okay. It's a theoretical stuff, so you might not be familiar with it, but just to let you know that the strategies of the ground theory approach exist. Okay. Okay. The last strategies, these are the five strategies that is mostly documented. So that could be some little thing else apart from these five, but the five are the most known. Okay. The last strategy is the desk strategies. Okay. The desk or secondary research. The easiest way that I could think of about example of this type of research is about history. That's what I think is probably most fitted with the desk or secondary research, because you can't do experiment with history. Okay, in science stuff you probably can do something about history, but history of this tree, history of this land by researching the carbon footprint, blah 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 blah. But there are other type of history like history, the situational history, what happened in history, and try to constructing the timeline about history. So in that way, they have to do a lot of desk research. They have to go places, finding documents, and then trying to make sense of what happened. Okay, I won't go into too much detail. We can talk about this later if you are interested, because it's sort of black and white thing about doing secondary research, and to explain more at this point. Okay, so what I think is that for our case, we mostly involve the experiment strategies. Okay, so that's why when you are preparing your experimental plan, we are talking always about what experiment to do. We are not talking about where we are going to survey or what case study we are going to do, that type of stuff. So most of us would use experiment strategy. But as a researcher, you need to know that there are other type of strategies for doing research that exist in this world. Okay. Okay, it seems like, okay. Now, since we are doing most with the experiment as our strategy, I will talk more about experiment as strategy and not going through each of the remaining four. Okay. So like I said earlier, that experiment as strategy is the most widely used form of research strategy in biotechnology. Okay, we probably cannot just do desk research and getting something out. I mean, doing desk research is probably just involving like doing literature review, but you can't, I mean, sometimes it's not, in our case, it's not acceptable somehow just to derive something and say that it is correct in our case, because for us the research problem might be different from other people. Okay, go back again to the conceptual design. So experiment is suitable for what? Okay, you are doing experiment when you experience with newly created situation or processes. Okay, this is very difficult to interpret things. What is newly created situation or processes? You might notice that whatever you do has already been documented. Okay, unless you won't be able to find any previous research when you are doing your literature review. So more or less whatever you do, because we were born like 2000 years too late maybe, so it's very difficult to find something newly created, but it is important. So to speak, you might have to find like something that your research is different from others. Okay, so if you experience with newly created situation or process, then you do experiment to verify it. Okay, the second point, that what experiment is suitable for is when you experience whatever that can be used to assess the effect of applied changes. What it means here is that if you more or less, if you want to see what factors or how these factors affect whatever you are doing, so that's when you do the experiment. For example, when you're varying pH, when you are varying temperature, when you are varying raw material type, that type of thing. So the changes that happen, if you want to see how it would affect whatever you are doing, then you start doing experiment. So in short, you are doing experiment when you have something new that no one else knows before. So we have to do something just to say that this is what happened. Another thing is that if you are interested in how one thing could change whatever you are doing, then you will choose doing experiment as your strategies. Okay. So once you know that you are going to use experiment, you need to understand the nature of experiment. Okay, so now we are talking about the characteristic of experiment. Okay, the first thing is experiment involving randomized. Okay. When you see the word randomizing, maybe you think about statistic. Okay. So what I talked what I really means about randomizing in this case is that your experiment, you should not stage your experiment. You should not stage your experiment. It has to be random enough so you can create errors. I mean, when I say errors in this case, is errors from some other thing. Okay, I mean, you can't do and get exactly the same exact result every time. So you are randomizing, and randomizing can be in anything when you are doing experiment. One way I used to give example about randomizing as your experiment is that when you take samples, or when you do the analysis. Okay, because sometimes if you notice, sometimes if you do a lot of experiments and then you have so many samples, sometimes you tend to analyze your sample by order. In that case, maybe you could have systematic errors and it won't be good. So the nature of randomizing has to be in the experiment. The second point is that in experiment, you should have as little outside influences as possible. What I mean here is that you should be in a controlled environment. Okay, you should be in a controlled environment. What I mean about in controlled environment means that if you are doing the effect of the temperature, so you vary the temperature, right? Because you want to see how temperature influences cell growth, for example. So what outside influence means in this case? That means that you have to have the same microorganism, have to have the same pH, have to have the same media and that type of stuff, so that you could control that the change that only happen from the factor that you are varying. So that means as little outside influences as possible. So you have to think about this in mind every time when you are designing any experiment, that what you want and what you should control. Okay. Another characteristic which is important is that the treatment group is decided solely by researcher. Okay, this seems to be a good point. Okay, because in experiment work, okay, you get to choose what you want to work with. Okay, unlike survey, or unlike population research, that you cannot really design the treatment group, what you want and what you want to fix. In case study, maybe you can just narrow down whatever group you want to study, right? But one main thing about experiment is that you get to decide what you want to use. Another point is that when you are doing experiment, this is a characteristic. So think about your research plan as well, okay? You have to have experimental groups and control groups in your experiment, okay? Think of control group as your benchmark, or whether it's positive control or negative control, you have to have the control group, okay? The experimental and the control group. This is very important because it will help you making like a conclusion, or when you are doing the analysis, you know whether whatever you do is changed or is not changed. Okay, depends on the control groups. Okay, so whether positive or negative control, actually you see it all the time. I mean, even for what they are doing today about the antibodies level after vaccination, okay, more or less they are like experimental group, and they have to have like a control. The control doesn't have to be like a group of people. It could be the beginning and the end as the experimental results. So choose wisely, okay? Just but control group is very important. And whether you need both positive and negative control, it depends on the experiment, okay? But choose wisely. If you get to do your experiment and you go to like the progress presentation for your thesis, sometimes you will ask whether this is when you have any control and how does it compare to control, and whether it's really changed from the control. Can you actually conclude that this thing really happens based on the results comparing with the control results type thing, okay? Another point about experiment is that you should always do the before event measurement to avoid false conclusion, Okay, before the event is that when you are doing your experiment and you expect that there will be some changes, you should have the initial values always. Always, you should have initial values always. If you are doing like fermentation, you should have time zero. It doesn't matter whether you are going to take sample every 12 hours or you are going to leave it for 72 hours and then take the final samples. You should have a beginning. Okay, time zero is very important. Okay, why do we need that? We need to do that to avoid false conclusion. Okay, to avoid basically false positive case. Okay, like what? Like say if you are doing, or actually I just have a recent example. I don't have the picture for you, but I could explain it to you. Okay, when you there was this case where they grow a bacteria on like a crude glycerol or something like that. Okay, at the beginning they take the OD reading, and the reading was about 0.2, and then they take samples along the way, and after say 72 hours that OD goes up to about like 0.4 or 0.5. Okay, the point is that if you don't have the initial 0.2, you might be misled that the OD of 0.5 is 72 hours. Oh, I have a lot of growth. Okay, but in this case, if you have the initial of OD 0.2, the change of OD 0.2 doesn't make that much difference. So you can't really confidently say that the cells actually grows in that source media. So this is very, very important thing as well. And talking about false positive, I think you have come across the words like false positive and false negative. Okay, if you understand those already, that's okay. But if you have heard but you don't know what it is, we can discuss about it later, but you need to ask me. Okay, maybe because I might forget the point. All right, so that is the characteristic of the experiment. So what I would like you to think about is that every time you are designing the experiment, okay, think about these 5 points of the characteristic of the experiment. In that way, you don't need to ask, do I need to take time zero samples? Of course you have to take, or you should know immediately that whether your experiment should have any control. Okay, so that is very important, and also this stuff that you should design your experiment such that you avoid any influence from other things that might affect the results that you get from your experiment. Okay, now let's see the good and the bad and the caution of the experiment. Okay, doing experiment give you a high degree of internal validity. Internal validity means that when you are doing experiment, and if you do it right, it gives you a good validity, or you can conclude with confidence that it works in the condition that you make in the experiment. Okay, that's called internal validity. So if you do the varying, for example, varying the temperature for enzyme activities, and you varying it from 30 to 60, and you find the best to be 55, you can say that is the best of 55, and you need to know that the best of 55, but you can't say the same thing for the temperature down from 30 or the temperature up from 60. Okay, it could have another peak to the left or to the right, you don't know, but you know for sure that during over this period, over this range of temperature, the best is this. Okay, it's you don't normally express this thing out, but you need to understand that doing experiment have a high degree of validity only internally during the range that you choose to do. Okay, the thing that you need to understand as opposed to high degree of internal validity is the external validity of the result. You can't assume. Okay, you can't assume that. You can't extrapolate. You can only interpolate. Okay. So basically the experiment works within the range that you choose. You can predict, but you can't confirm. Another down thing about the experiment is that the outcome or the result may not be correspond with actual conditions. Okay, this could be a lot of problem. That means you, for example, if you are doing experiment and trying to use like a type of raw materials, and you do things in flask and in fermenter, and then you are upgrading and you are and maybe some companies or some factories wants to use the method or whatever you do in the real situation, the result might not be exactly the same. Okay, so that's why when you are doing experiment and you are going bigger scale or you are going to real world situation, it could be different. So the result that you get from the lab work might be different, and if you want to make the real world situation, normally the real world situation is worsen than whatever in the lab, because in the lab you can control everything, right? In real world situation could be different. So that still needs some process adjustment when you are using your research with the real world situation. This is very classic. Like say for vaccines, like COVID vaccines, okay, they have phase 1, phase 2, phase 3, and at initial stage, it's mostly like a lab work, a control group of people, but in real world situation, it's different. So you could see, I mean, if you read some news, you probably see that, hey, this vaccine, when they are using in real world situation, is not like in phase 2 and phase 3. So that's how it happens, because in actual condition or in real world situation, you will lose the ability to control other things that could influence your results. So that is something that you have to bear in mind. Okay. Another point, okay, is the caution for the experiment as strategies is that is not all the interferences can be covered by experiments. This point actually reflect that when you do real world situation, the real world situation or actual condition, you might get a different result. Okay, the result may be good, but not as good as in the lab. Okay, because in real world situation, there are so many things that could happen, right? For example, like in a okay, just go back to the vaccine case. It's easy. It's easy to explain. In real world situation, you can't control the age of the people, that the physical condition of the people. You can't control the disease or the condition of the disease of the people that get vaccinated. Unlike when you do like phase 2, phase 3, it's a small group and you can actually select it, the subject that you want to do the experiment with. Another point is that if related to our field, you are doing experiment in the lab, you could control contamination, you could control many things. You could even control the raw materials that you use. But in real world situation, everything varies. So all interferences cannot be accounted for in experiment. So this is something that you need to understand. Okay. So I mean, if you encounter, if you talk with someone and they say, "Oh, my lab result is so good," but it doesn't work when you go to use the I mean in the real situation. So, but I mean that doesn't mean that you don't need to do some study before you actually use it in the real world situation. You don't go straight away to real world situation. But if you know that there are other interferences that happen in real world situation, that is when it will help to improve the result in real world situation to be as close or as good as in the lab. So it's about knowledge. Okay, that was about research strategies. Okay, I hope you get a little bit more sense about doing the experiment, a good thing, a bad thing, and the thing that you need to consider about doing the research experiment. I mean, the experiment, because the experiment is not like the final answer. Okay, it's just a strategies for us to obtain something. The next point in technical design, okay, is the research materials. So research materials, let's see. Okay, when we talk about materials, it's not just a material method. Okay, materials is actually something that you is touchable. What include in materials when we talk about materials in research? Of course, it includes the method. Okay, because one section you always have materials and method. Okay, now you have to think deeply about the word materials and method when you actually write down your materials and method. Okay, so research material involve, for example, methods, lab supplies. Lab supplies means what? Means your flask, your bottle, your chemical, your pipette tips, your microcentrifuge tube, your volumetric flask, your glassware, your plasticware. That is the lab supply. You have to think about that when you are thinking about research material as well. And as chemicals, that's what I said already, but you need to think about it very carefully also, because you have chemicals that you use for your cell growth. You have chemicals that maybe you need for preparing your raw materials. You have chemicals that you need for analysis. Okay, you have chemicals that you probably need to use with your other instruments as well, the solvent and that type of stuff. So that is the chemical. Chemical is not just what am I going to use for fermenting myself. But instrument, everything. Okay, everything that is not what I said above. Vortex mixer, centrifuge, microcentrifuge, large centrifuge. Do I need to use that? GC, HPLC, columns. I mean, GC, HPLC columns, what type of column, what else? You need pumps, you need blah, 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 blah. You have to think all that when you're actually designing your research, okay? Don't just think, okay, I'm going to do centrifuge. What centrifuge are you going to use? Hey, we don't have access to that centrifuge. What are you going to do? Okay, so you have to think through this before you actually start doing your experiment, unless you will be like, wow, I forgot to think about this. How could I use this thing anywhere? Okay. Another thing is funding. Funding is sort of like I told you earlier that maybe you don't have to think about it at the moment, but when you actually start doing your own research, okay, or you happen to have to do your own research, you have to think about where you get the funding from and how much money do I need to finish off everything, blah, 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 blah. So that is another type of plan. Okay. Now, to emphasize a little bit more about the research materials is that when you start designing your project, you have to think what kind of material is needed. So what method do I use? What glassware, what chemical, what instruments? Okay. So what do I need? Another equally important question is how do I get them and where do I get it? Okay. How do I get it? Do I need to buy it? Or I can borrow from someone, or I can use whatever it is in the department already. Or if it is instrument, where do I get it? Like say, for example, if you have to use the large centrifuge, do we have it? Or if we don't, where am I going to use it? If you want to do something like freeze drying or spray drying, where am I going to use it? How much is going to cost me to use it? That type of thing is something that you need to think about. So whatever you write down in your research proposal, in your materials and method, you have to think about, okay, I'm going to do this and where am I going to do this and what instrument I need to do this and how I'm going to obtain those instruments, or I have to borrow someone or I have to go somewhere to do it. Is it doable? That type of stuff. Very important. So much. Okay, so much. Because if you think about this, when you actually start doing things yourself, you turn your thoughts into the actions. I mean, unless it will be just be your thought, okay, because you don't have something to action on. And of course, it's some theory into reality. So these questions are very important. Think about it every time you write down something, every time you write down centrifuge or cell separation, every time you write down filter it through this and do this and do that, think about what chemical involved, what instrument is involved. Do I have it? Do we need to buy it? Or do we need to go somewhere else to use it? Okay. And is it accessible? Because in our university, we have almost any kind of instrument or like very fancy machine, but sometimes it's difficult to get through to use, or sometimes they don't accept your sample. I mean, your type of samples, that could be possible. So it is something that you need to find beforehand, whether it is possible for you to do. Okay. Very important, because if you don't think about it, you get stuck. Okay. And once you get stuck, after you do your proposal, it's very difficult to continue, because you already think many things ahead of you already. Okay. Now, in this slide, I want to talk about the thing that happens when you are selecting materials. So when I talk materials, okay, you think about the previous slide, okay? There's so many things that involve in the word materials. Important things about selecting materials is the difficult part is that if you know a lot, there will be a lot of things to choose from. Okay. The point is there's a lot of things to choose from. I mean, like example I gave yesterday, or your friend asked yesterday. If you want to analyze one thing, or you want a method to do something, there are so many available. Okay, there's so many available. That is one good thing about our generation. We have a lot of people doing a lot of things, and we have a lot of things to choose from already. Okay, but it could create difficulty for you, right? Like just say, I want to analyze glucose. There are so many ways, right? But the point is, before you go into that stage of having so many things to choose from, you have to be resourceful first. When I say you have to be resourceful, is that you have to have enough knowledge and you have to have enough resource to come up with interesting materials. Okay, that's why you have to you have to be knowledgeable. That's what I'm going to say. You have to be knowledgeable if you want to do the cell disruption, the cell breakage. If your product is internal, is intracellular, if you want to break the cells, you have to have like tens of ideas of how to break the cells. And then you narrow down all the possibility whether we have that instrument or it is better this way, and whether this way is if you destroy my product if I use it this way, or this type of thing. You need to come up with it. You have to be resourceful to be able to come up with interesting material. Okay, sounds difficult. It is difficult. That's why sometimes reading a lot will help you, because it helps you see so many things. And sometimes actually going to a seminar or going to progress report and listen to your friends, sometimes it gives you idea as well. I mean, I can say this 100% because when I go to your seminar, like your student seminar and also your progress report, I sometimes gain some idea from what other people do. Okay, so you have to be resourceful. Another point I want to talk about selecting the materials is that a good thing is you have freedom in choosing. Okay, point is you have to be resourceful to come up with something. That's when you are having a lot of things to choose from. And the good thing is that you have a freedom to choose. Okay, you have a freedom to choose based on whatever is you need. Okay, the hard part is making choice. Okay, hard part is making choice. Maybe it is as hard as finding what to choose from. If you have too many, you have to be able to make the choice. How would I make a good choice? That's what your friend asked yesterday, right? You want to do the same thing. You have two methods. How do I know the two methods? Which one is better? I mean, it depends. It depends on so many things, right? You see, it depends on the materials that you have. It depends on the instrument that you have. It depends on your skill, the skill that you have as well. But at least you get something to choose from. That comes to this point. You have to have the idea about advantage. Okay, advantages of the materials, the disadvantages of the material, and the possibility of options that are available. So all of these things involved in selecting materials. At the end, you want to be resourceful enough to come up with many choices and then have knowledgeable enough to know which one is good, which one is bad, and be able to select the final material, each of the materials that you made. Okay, you have to think about it. The point is, what if I have everything, but I don't know how to do it? Always you could ask for some helps. Okay, I mean, if you want to use HPLC or GC and you don't know how to use it, but you know it is the best thing for you, and you do know a little bit about it, but you don't know exactly how to use this specific brand and that type of thing, people can help you. Okay, but if you have zero idea, you don't go for help. You have to educate yourself first before going to help. Okay, so that is about selecting materials. Sounds hard enough, but it is something that is important. Okay, it is something that is important because it is a large part of your work. Okay, it involves the success, the delay of your work as well. The last point about technical design is research planning. Now we are talking about the planning in itself. When you plan, okay, for the previous topic, okay, we talk about the strategies already. We know that most of us use experiment, and when we design experiment, we have to think about the characteristic of the experiment. Then we thought about the materials. Now it's about planning. When we talk planning, we talk about 4 things, okay? What, how, when, and how much. Okay. Like I told you earlier, many times that you might not be aware or have to be like very serious so much about how much, because it's your advisor who find the grant, who find the money for you. Okay, you might worry about your own expenses, but in doing your research, your advisor is fully responsible to finding you the money to do your research. However, I need you to think always that even though your advisors is finding money for you, you have to think about how much that you use for yourself as well. What I mean is, it is in the point that you don't fail the experiment too often. Okay, you have to learn from your mistake, but not making mistake all the time. That is something that I want to tell you. Okay, now let's see the what, how, and when. Okay, when you're planning your research, okay, you think about what first, right? It is your research objective, your research question. That is what you have already been covered with. And the how is the research strategies and research materials that we talked about earlier. Part of the planning really is the when and how much. This is the planning part. Okay. How much is what we talked already. The when is the research timeline and the activity schedules. Okay, if you're looking at your proposal report, it is toward the end of your proposal reporting. Okay, I know that a lot of people don't take the timeline seriously. Okay, but actually I was the one who actually don't care so much about making a good timeline, but I learned over the period of time that think carefully about the timeline and stick to the timeline will get you to finish everything as you plan. For example, you have 2 years, okay? You have 2 years, so that is 4 semesters, and that is 24 months. What do you have to do for your research? You have to start from the beginning, okay? Doing this, doing proposal, blah, 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 blah. And at the end of 24 months, at the end of 24 months, you have to finish your thesis defense already. And the best thing is you already have your manuscript for submission already. So if you don't plan well and stick to your plan, everything couldn't be done. Okay, I know a lot of people think that I really want to finish my degree in 2 years, but a lot of people don't get it within 2 years because they don't stick to their timeline. Okay, and making a good timeline is really, really important, I can tell you. Okay, it will help you so much in planning your study. Okay, so what you should do when you're planning your timeline and schedules, okay, the first thing is list out all activity to be carried out. Okay, list all activities. Okay, don't be too shy with having so many activities. If you can list out 100, so that's 100, okay? And I think your advisors will help you grouping them and scale them down. Okay. So the activities to be carried out, mainly if we talk about doing research, is the experiments based on your research plan to achieve the objective. See, you do the experiment to achieve the objective. This is something that you have to think always, okay? So you list out your experimental plan, okay? You list out your experimental plan that, okay, I'm doing this experiment, this experiment, this experiment. Then you have to think, this will help you with making more effective research, is that thinking about the result that you could obtain from each of the activity that you listed out. So basically, what results you expect from each experiment, okay? And with expected results, it will give you the idea of what data you are going to collect and what analysis will be involved. And when I say expected results, means that if you are doing one varying something, you expect that you should get a best result with this pH, blah, 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 that type of stuff. So it's sort of like forecasting things that would happen when you actually start doing your experiment. After these 2 steps, you should sequence your activities, okay? Decide if this experiment to be done, have to finish this one before starting that one, or this one can actually do a long way with that one, or this one can do like halfway and then you can start another thing, okay? And then you fit that into your timeline of activity, okay, which is a time frame of each experiment. Okay, the best thing is to give you examples, okay? This graph, okay, this chart or this table that you see is a very popular way to do your planning, to do your research planning. It is called a Gantt chart. Okay, this is the name. Okay, this is the name, the Gantt chart. So in the Gantt chart, what you do is see that you reach out the research activities. Okay, you just put in your research activity, but expected result and blah, blah, blah should be in your research plan already. Okay, research activities, like for this for this research, it is actual research that has been done. This is the timeline of the master degree research that I supervise. Okay. So the activity is listed out into 7 main activities. Okay, 7 main activities. The first one is preparing the cells, the biomass. Okay, and then doing the treatment of the biomass, prepare hydrolysate, doing ethanol production. Another experiment here, preparation of manuscript and writing the thesis report. Okay, everything is summed up within 12 months. Okay, you will see that at first 1.5 months for preparing materials, but you don't have to wait until the material preparation is finished. You can do some studies already, right? So you see the half-month overlap here. So that means that during this month you do 2 things at the same time. Okay, but the third activity cannot be done until the second activity is finished. So it has to be in series, not in parallel. But for preparation, you don't need for it to be done before you do the fourth activity. So you can overlap these parts. So you see that with this work, they're preparing the hydrolysis for the fermentation for half a month. And then after they have like a good amount of hydrolysis, then you can start on the studies here. And the next one cannot be done before the fourth one finished. So it has to be in series. Okay. And when you get everything, actually these students could prepare the manuscript before then, because for the manuscript, you can write like an early results first also. Okay, preparing the manuscript can be done together with preparing thesis report. Okay. What I'm suggesting for every one of you is doing a very, very good proposal. That because the first 3 chapters of your thesis report, most people will take it from the proposal. That means introduction, background, objective, literature review, materials and method. These 4 things contribute to the first 2 or 3 chapters of your thesis report. If you do well for the proposal, you just take that into a thesis report. If you don't do well and you just do it and just let it be, you probably have to write a lot, especially in materials and method, okay? Because it needs to be updated every time, because you write something in your proposal, when you actually start doing something, is feedback loop. When you are actually doing something and you change something, you have to change your materials and method as well. So this is how the Gantt chart look like. And like I told you, that if you plan out well and stick with the plan, you finish within due time. Okay. And for those of you who is actually keen on planning, okay, you can have another Gantt chart for each of the activities. Like for this one, you have like 3 months. You can have another little Gantt chart for that 3 months, that this week I'll do this week I'll do that week I'll do that. It can go on forever, down until the day, you know, this day I'm going to do this day I'm going to do that, right? That's a Gantt chart. Another type of planning, okay, you don't see this a lot, but some of the grant application, I talk to your future that if you need to write some grant, and they probably want this type of activity tables. Okay, it can accompany the Gantt chart as well. So the activity and output tables, okay, it is similar things. You have research activities that you listed out here, but instead of having like the arrow showing the length of each activity, you actually set the duration. Okay, you see that sometimes the duration the time that you are going to do this thing are overlap, like this 1 and 2, they overlap on February. So that means that in February you are doing 2 activities together, and also 3 and 4, just like what in the Gantt chart. What is extra in this one is expected outputs. Okay, this sort of table is a good way for you to keep track of what you will get at the end of each of your activities. Okay, like for activity 1, expected output is that you have to obtain chemical and biomass for experiment. If you don't get that, that means that you are not reaching your goal. Okay, research activity 2, you do experiment and you expected to get that. The numbers that you get is temperature, time, and percent acid for the treatment, and you also want to know the sugar and ethanol that you get. Okay, in preparation, even though you said you prepare something, you have to have some aim. Okay, you have to have aim that after you prepare these, how much of the thing you will get. For example, preparing hydrolysis for ethanol production study, enough hydrolysis for fermentation, about 1 liter. That means that at the end you have to get 1 liter. It's not like, oh, I do it, I prepare it, but I don't know how much I'm going to get. The point is that if you know how much you get, you know how much you have to prepare. This is about materials again. Okay. Then study on ethanol production. You have to have some expected results here. Like preparing manuscript is not just like preparing manuscript October, November, and at the end of November, I still preparing my manuscript. I have already prepared 50% of my manuscript. I finished introduction, I haven't finished anything else, cannot, okay? So expected output, if you're preparing manuscript, is that you should have a manuscript draft ready for submission, or at least first draft or whatever, it depends, okay? But you have to finish it. Okay, preparation of thesis report. In this one, say version ready for exam committee. Okay, may not have to finish the defense yet. Okay, and when you said, oh, another thing I want to focus is that when you do the research activities here, it's not doing just experiment. Okay, research activities start from preparing the experiment, doing experiment, taking samples, and making analysis, and have the data, have the results. Analyze the results. If you have to repeat, you have to repeat within that time period that you set out. That is important, okay? Because I see a lot of students have like, okay, I do this, I finish the experiment. Have you analyzed? No, not yet. So it doesn't work that way, okay? To make it efficient. Okay, I believe this is the last slide, okay? I want to add it here so that you have some idea about these words, okay? They are part of research planning, and maybe in the future, if you write down some proposals, is the word output and outcome. I think that you have heard it before. Output in Thai is ผลผลิต. Outcome is ผลกระทบ, I think. Okay. So basically, if you look at the right hand side here, okay, outputs enable us to fund outcomes, but without outcomes, there is no need for output. I got this thing from this website, and I think it gives a lot of sense. Okay, so outputs enable us to fund outcomes, but without outcome, there's no need for output. What it means is that, you see, outputs is the what you get from your research. Okay. In general speaking, output is, if you have a company, you produce product is your output. if you are a phone company, you provide service. So service is the output. Okay? You have company, you have profit, you have revenue. That is the output. It is something that you could touch. Okay, it is something that you could touch. Outcome is the why. The why is that what does it create? What is the meaning? What is the relationship? What is the difference? What does it make to? What does this thing makes change? For example, what could be an example? Okay, for example, researcher who do like photovoltaic, solar cell, photovoltaic, they do, and the output of their research is the product, which is a solar cell that could generate electricity. You can store electricity you can store the power in the battery, and then you use battery and you use the power from the battery. That the solar cell is a product. The outcome is really the thing that this product could change. Okay, that means for this example, it means that people who doesn't have the power line have access to power, have access to electricity, have access to electrical things like lighting, computer, or refrigerator. So that is the outcome. Okay, so that's two things. That's two different things. Okay, output is something that you could actually touch. Outcome is something that the output makes this. Or for the service, right? The output of service, for example, in a business like hotel, the hotel business is the service business. It make profit, it make revenue, but the outcome that the hotel business has is that is one of the main business in Thailand that draws a lot of people to come to Thailand before COVID and generate a lot of work. A lot of people get the job, and that type of thing is the outcome. Okay, I'm not going to give like examples that directly to us, but I'm giving this example for you to think about your own research. What would be the output and outcome of your research? Okay, if you could get output and the outcome right, it helps you answering back to your research problems. Okay, and the reason to do your research. All right, and I think that's it. Okay, I have a little bit of bibliography for this chapter. That's not a lot. Basically, it's half that I get some contents from, and the rest is the experience that I want to share with you. Okay, and that actually ends my lecture. So from now on, I think I'll take any question if you have any question, or if you want me to explain a little bit more in some of the slides, or you have something to discuss, something that you want to ask, maybe. Nope. I thought there would be more questions today. Any question or anything? You can ask me about your own research, about what you are doing right now, and maybe I could give you some suggestion that relates to our lecture. มีใครอยากถามอะไรได้นะคะคือที่เกี่ยวก็คือ เป็นเนื้อหาในเลคเชอร์ก็ได้เนื้อหาไม่ใช่ในเลคเชอร์ก็ได้แต่เกี่ยวข้องกับเรื่องการเตรียม proposal ของพวกคุณหรือว่าเกี่ยวข้องกับอะไรพวกนี้ของพวกเราอะค่ะได้ได้หมดนะคะไม่มีไม่มีเลยโอเคค่ะโอเคในถ้างั้นในระหว่างนี้เนาะเดี๋ยวอาจารย์พูดถึงว่าครั้งหน้าที่เจอกันเราจะทำอะไรกันบ้างอะนะคะเอ่เดี๋ยวอาจารย์ขอเปิดเอ่อมีแป๊บหนึ่ง ครั้งหน้าที่เราเจอกันจะไม่ใช่เลคเชอร์นะคะแต่จะเป็นการทำกิจกรรมเนาะเป็นการทำกิจกรรมซึ่งอาจารย์ขอเปิดนี่แป๊บหนึ่งก่อนนะคะโอเคค่ะก็คือจะเป็นการทำกิจกรรมนี้นะคะเป็น practice activity assignment นะคะเราจะเห็นว่าอาจารย์มีเปเปอร์สำหรับ practice activity อาจารย์ไม่ได้อยากจะให้ไปอ่านเปเปอร์ทำความเข้าใจก่อนนะคะอาจารย์ไม่ได้ให้จะไปอ่านเปเปอร์นะคะแต่ว่าถ้าเราได้ดูล่วงหน้าก่อนนะคะเราก็มีถึงเวลาจะได้ถามอาจารย์ได้นะคะว่าที่เข้าใจตรงนี้ถูกไหมนั่นนี่นู่นนู่นนี่นั่นอะไรยังไงสิ่งที่เราจะทำกันในห้องนะคะอาทิตย์เราเจอกันวันจันทร์บ่ายเนาะก็คือเราจะเอ่อเราจะดูเปเปอร์นี้นะคะเราจะดูเปเปอร์นี้แล้วเราก็จะศึกษาจากเปเปอร์นี้ว่า กลับไปแล้วเนี่ย planning เขาเป็นยังไงนะคะถ้าเรามีเวลาอาจารย์อยากจะให้ไปดูนิดนึงก่อนจะได้พอรู้ว่าจะทำอะไรบ้างนะคะอาจารย์มีแบบฟอร์มสำหรับ assignment เนาะแล้วอาจารย์ก็มี class example ซึ่งเราสามารถไปดูล่วงหน้าก่อนได้นะคะหลักๆแล้วเนี่ยอาจารย์ก็คืออยากจะให้เราได้ฝึกดูอะค่ะคือคือตอนที่เราทำ proposal เราอะเราก็คือจะต้องคิดของเราเองนึกออกไหม ว่าว่ามันจะต้องทำอะไรยังไงแต่สิ่งที่อาจารย์จะทำใน assignment นี้ก็คือมีเปเปอร์ที่เขาทำการทดลองเขารายงานหมดทุกอย่างอาจารย์อยากจะลองให้เราแบบ extract มาว่าไอ้สิ่งที่เขาทำเนี้ย objective แต่ละมี experiment อะไรบ้าง objective เป็นยังไงบ้างเขาเก็บ data อะไรสำหรับแต่ละ experiment ในลักษณะพวกนี้นะคะพอดีที่อาจารย์อันนี้คือสิ่งที่จะทำเนาะ เพราะฉะนั้นก็คือในชั่วโมงหน้าเนี่ยอาจารย์จะไกด์นะคะว่าเราจะทำ1 2 3 4 5แต่หลักๆแล้วก็คือเป็นการหลักๆแล้วก็คือเป็นการเป็นการนั่งทำงานนะคะนั่งดูตรงนี้ด้วยกันโอเค Epic what I just showed you just now is the paper for the class activity. Next week, we are going to see each other again on Monday afternoon, right? So what we are going to do was we are going to re-engineering, sort of like analyzing what that paper does for that experiment. Because basically, when you are doing your own proposal, actually you have your proposal exam already, right? Epic?
Yeah.
Maybe this assignment is easy for you at the next assignment as well, okay? But basically, it's just think of, even though you have already done it, just think of it as a practice or something. Just want you to figure out each point of the work that they do, like say how many experiments they do for each experiment, what are their objectives, what do they do, the materials that they use, and that type of stuff. So it will help in preparing the proposal. I mean, by doing this assignment, is probably help you to sort of having a more thought onto your own proposal somehow. Okay. you can go and have a look in Google Classroom first right ไม่มีใครถามอะไรเนาะ okay so I guess you don't have any questions so next time that we meet if you could see the thing before that I show you before class it would be great and I'll leave the activities during the hour and yeah let's see how it goes alright ค่ะถ้าเป็นแบบนั้นก็ขอจบการเลคเชอร์ของวันนี้เพียงเท่านี้นะคะแล้วเจอกันวันจันทร์บ่ายเนาะ Okay I'll finish my lecture for today and I'll see you on Monday afternoon Okay ขอบคุณมากค่ะทุกคน
Thank you อาจารย์
ค่ะสวัสดีค่ะ
ขอบคุณครับอาจารย์สวัสดีครับ
ค่ะ